Hi guys, today I'm going to be talking about COVID-19, its effects in the body, the vaccines available, and its origins. So, the naming SARS-CoV-2 is short for Severe Acute Respiratory Syndrome Coronavirus 2. The number 2 in the virus's nomenclature is due to COVID-19 being a sister virus to SARS-CoV, the primary viral isolate defining the species. The name SARS refers to the classificational relationship between SARS-CoV and SARS-CoV-2, COVID-19. SARS-CoV-2 belongs to a larger species of viruses, however, called SARS, and unlike some of its virus sisters, SARS-CoV-2 is not, according to the newest research, is most likely not man-made. The origins of SARS-CoV-2 are still a bit unclear, however, but according to the CDC, it was most likely transmitted to humans through a species of bats called Rhinolophus in Wuhan, China. It is highly speculated that the disease had the pangolin as an intermediate spreader between the bat and the human that first contracted it. This most likely happened in the Wuhan wet markets, which have already fostered an environment for SARS before. However, since SARS-CoV-2 is not zoonotic, meaning transmittable from animals to humans, according to a famous UK study, the origins of the disease are still very vague and unclear. So, SARS-CoV-2 is an RNA virus that has a viral envelope protecting the virus when it is not within a host cell. The envelope structures the proteins that the virus needs in order to infect other host cells. The other parts of the virus are envelope, spike, and nucleoproteins. So instead of a capsid, COVID-19 has these nuclear proteins that allow it to maintain its structure and replicate effectively. Furthermore, spike proteins allow the virus to latch onto new host cells and break them open in order to infect them. And finally, envelope proteins help the virus replicate and once it infects a new host cell. So here I have a replication of COVID-19. So the long black toothpicks, those are a representation of the S proteins, spike. The red short toothpicks, those are a representation of the M proteins. And finally, the blue toothpick is a representation of the E proteins. So this is what the surface of a COVID-19 virus looks like. The model. SARS-CoV-2 targets cells in the lungs, nasal passages, and intestines in order to initiate the COVID-19 disease in human beings. So COVID-19 enters host cells by binding to the host cell's receptor enzyme ACE2 and TMPRSS2 through its receptor binding domain, and it is proteolytically activated, meaning it's activated through peptide cleavage by human proteases. So COVID-19 is a lytic virus, meaning that when replication takes place in the lungs, for instance, COVID-19 damages respiratory cells along the lungs tracts. Specifically, when the spikes of SARS-CoV-2 bind to the receptors mentioned early, it's activated proteolytically, which then causes the viral envelope to merge with the membrane of our own cells, allowing the virus to release its genetic material in the inside of a healthy host cell. So the genetic blueprint of COVID-19 is RNA, which acts as a molecular message instructing our host cell's machinery to read the template and translate it into proteins that make up new virus particles. After the viral spread of these proteins, COVID-19 leaves the host cells via exocytosis. So, there are three main variants of COVID-19 in the US. There's P1 from Brazil, B1351 from South Africa, and B117 from the UK. However, it is speculated that there may be more. But since we don't have a lot of research on these new variants as of now, those are the three main ones that the CDC is talking about. So these variants mainly differ from the wild type of COVID-19 by having mutations to these spike proteins, and this mutation makes the variants more. They make them bind to our host cells more tightly. And that's why they're considered more contagious. Current research shows that the two vaccines available in the U.S. will still work effectively against these variants. They'll just be less effective. So this variant occurs when a new virus has different RNA from its parent virus through a mutation, like adaptable mutation, and a strain is a type of variant where the new virus has different RNA from its parent through a mutation, and where the new virus has different physical features from the parent virus. Okay, so unlike the conventional vaccines in the past, where an unactivated pathogen is introduced into our body's host cells in order to trigger a response, the Pfizer and Moderna vaccines use a new revolutionary method in which mRNA is delivered into specific host cells in order to provide general instructions for our immune system to temporarily make specific viral proteins that trigger an immune response. This immune response is actually what causes the body to make the antibodies that will fight COVID-19. So in the context of the COVID-19 vaccine, T and B cells recognize and respond to viral antigens by producing many protective reactions and effector molecules. This means that the T memory cells help cause immunity that outlasts the antibodies from the vaccine. They target infected cells and stop the replication caused by SARS-CoV-2. On the other hand, B cells help initiate the antibodies that fight COVID-19 directly, rather than targeting the infected cells. And naive T and B cells are stimulated, and they differentiate into effector and memory T cells and B cells. So here I have a diagram to show this. So here we have naive T and B cells. So these have not differentiated yet. When the vaccine is introduced, the B cells, they start producing antibodies that will target SARS-CoV-2, and T cells, they will send certain molecules that will target cells that already have COVID-19 and cause them to go into apoptosis, which is cell suicide. So that's why these two are very crucial in the COVID-19 vaccine context. And it is believed that the T cells will actually outlast, this effect will outlast the antibodies created by the B cells. However, the research is still not very sure as to how long immunity lasts. And finally, if COVID-19 is introduced to the body after vaccination, antibodies triggered by B cells will target the virus and most likely eliminate it. All right.